Hi there. In this section, I'll be talking about multi-gene families and multicellularity. Multi-gene families are important to the development of multicellularity, and those two will sort of be woven together in the next lecture or two. I'll talk about first gene duplication events and their importance in the formation of multi-gene families. And then I'll discuss specifically some multi-gene families and how they have evolved. Here's a list of multi-copied genes which form families. By no means is this an exhaustive list. So at the top you can see actin genes, where there are at least 61 versions. Alpha and beta globins, where there are 1 to 5 copies each, and sometimes many more than that. The histone genes, at least 75 different versions of those. Lacases in fungi, and ribosomal RNA genes, which I've already talked about before, 1 to 23,000, and in some cases even more copies than that, and many, many other gene families. Most multi-gene families begin with a single gene, and then there's a gene duplication event, usually from a crossover, a transposition event, or something similar to that. So starting with one gene, let's call it A, you can get a duplication of that gene so that now you have two versions of A and A prime, and as long as the one version of A retains its function, then A prime starts to mutate and form a different gene over time. This process can continue through more crossover events, transposition events, etc., generating more versions of the gene family and more divergence, such that some members can be quite different from the original gene. You can also have genes move to other chromosomes, and there's often generation of pseudogenes. And the pseudogenes are usually of two types. One is a truncated gene that then is not expressed in any way. But there are expressed pseudogenes that ultimately result in a protein being formed, but it's a non-functional protein. One very large multi-gene family consists of genes that produce flagella in bacteria. And these are important for motility in moving the bacteria towards food, away from enemies, etc. And so some bacteria form flagella only at one end, some form flagella at both ends, and there are some that form flagella throughout the length of the bacterium. This slide actually has quite a bit on it, but if you start at the upper left, you can see a model of how the flagellar apparatus is attached to the inner and outer membrane of this gram-negative bacterium, such as E. coli. So the basal apparatus is integrated and anchored into the inner and outer membranes, and it also traverses the cell wall. On the outside of the cell, then you have the hook and the filament. The little inset there is an electron micrograph. Negative staining is used there to actually visualize one part of this apparatus. The diagrams in the lower left show a few different configurations for flagella in these bacteria. One is on the far left. It's just hooked onto one end of the bacterium, and then you can see others where you have multiple flagella and flagella at both ends, and flagella pretty much over the whole length of the organism. The figure on the right is a model of the flagellar apparatus showing which genes and which proteins make up each section. And these are all members of the same multi-gene family. In fact, you start with a very small number of these genes, mainly in the base of the flagellum, and then you can build out from there. And the evolution of these, as well as how they're put together, follow the same pattern. So looking from the inside, then things like fly N, fly M, fly G, fly F in the C and MS rings appeared very early in evolution and weren't associated with a flagellar apparatus. They were actually part of a secretion system. And then as you work out further and further to the flag H, flag I rings and outer membrane, those appeared later in evolution and functioned in a slightly different way. And finally you get to the hook genes and proteins, and to the filament and filament cap, which appeared later in evolution. When the arrangement of these genes in the genome is examined, bacteria have different clusters of these genes, all related, but the ones that are within a cluster are generally more closely related than those from a different cluster. In this case, there are 7 gene clusters, and if you look at the top one, there's flag A, flag M, and then a hypothetical protein, which is similar to flag A and flag M. And then there are other clusters as well. As I mentioned earlier, these genes often move to different locations in the genome or onto a different chromosome. And that's what's happened here, that the genes by recombination or transposition or some other means have moved to different locations in the genome, followed by further expansion of the gene family in each of those locations. Assembly of flagella has been studied extensively, and it's been found that it's built from the inside outward. And if you look at the ages of the genes, they follow the same pattern. So you start with the basal ring up at the top left, and if you go down that first column, that's built up, and then you get a second collar built to go through the second membrane, and then finally you start forming the hook in the middle top there. So the top is formed, and then finally there's multiple units, protein units that come out to form the filament, and it's just repeated over and over and over again. If you go to this website, YouTube, you can find an animation of this that shows the same thing. It's really actually very interesting to see how these things are built up. On the right, you can see the basal assembly at the bottom going through the first membrane and then proceeding through the second membrane, up through the hook, and finally up into the filament. And that's exactly the pathway of construction and evolution of these genes. This diagram represents the proposed evolutionary pathway that led to flagellar apparatus, and you can see it starts from the inside membrane, the inner membrane, as a pore. Part of these also became the parts of the ATP synthase molecules or complex, and then became a secretion system where now there were portions that were on the inner membrane and other portions that were on the outer membrane for exporting particular chemicals. Part of these became important in surface adhesion, that is, adhesion to one surface or one bacterium or another, and then an adhesive pilus, with a cap, and then a pilus cap rod, all containing ATP synthase and ATPase as an energy producing mechanism. And then the final step is to produce a flagellum. In this case, the ATP synthase was not used, but a different system. The moat A and moat B proteins were used as the motive force to propel the flagellum. Archaea also have flagellar apparatuses, and on this figure you can see how those are distributed among the archaea. In the middle you can see M or NM, that means modal or non-modal. So the ones that are modal also have flagella. As in bacteria, archaea also have the flagellar genes located in clusters, usually one cluster, and the genes are members of a multi-gene family. Some are more related than others. But you can see in the different groups of archaea, the arrangement of those genes, as indicated by some of the colors, are similar in some cases. Another multi-gene family that's been investigated a great deal is the globin gene family. And they've been investigated all the way from fish up to placental mammals. And there's actually a very interesting evolutionary story in that. So on chromosome 16 in humans, there are 5 globin and globin-like genes, including alpha 1, alpha 2, pseudo-alpha, pseudo-zeta, and zeta. And then on chromosome 11, there are beta and beta-like genes, including epsilon, gamma G, gamma A, a pseudo-beta, a delta, and the true beta. The gene expression of these differs depending on the developmental stage of the organism, in this case human. So very early on, zeta and epsilon, the alpha and beta type globins, are turned on to the greatest extent. And then by 3 months later, they're almost non-existent. During that time, then, the alpha is expressed, and the gamma globin is expressed as the beta analog. And then slowly, the beta, the true beta globin is expressed, the gamma decreases, and a little bit of delta shows up just before birth and continues upward to a few percent during the first 6 months of life. The different hemoglobins, the different globins in general, are able to pick up and release oxygen at different partial pressures of oxygen. And so the adult hemoglobin picks up oxygen only at higher pressures, partial pressures of oxygen in the blood, whereas the fetal hemoglobin can pick up oxygen at lower concentrations of oxygen, so that you can have a net movement of oxygen from the adult hemoglobin to the fetal hemoglobin. The same with myoglobin. The fetal hemoglobin and the adult hemoglobin can give up, essentially, oxygen to the myoglobin in the cells. So there's a net movement from the adult hemoglobin to the fetal hemoglobin to the myoglobin in the cells. This is very important in placental mammals because the oxygen has to move into the mother's blood from the lungs and then be transported through the mother's blood to her own tissues. But the oxygen also, there has to be a net movement of the oxygen from the mother's blood through the placenta into the fetus's bloodstream, and then to the fetal tissues. So there has to be a net movement that way, and the only way that that can be done is by having these different globins and hemoglobins. Here's a different way of looking at it. So the oxygen moves into the mother's lungs. The oxygen is picked up in the mother's blood by the adult hemoglobin, which contains 2 alphas and 2 beta subunits. And then that oxygen is transferred into the myoglobin of the mother's tissue. Also, the oxygen is transferred through the placenta to the embryo or the fetus's blood. So in the embryo, you have zeta and epsilon subunits, and in the fetus, then you have alpha, gamma globins. And those pick up oxygen better than the adult hemoglobin alphas and betas. And so there's a net transfer there, and then ultimately that's transferred to the embryo or the fetus's tissues into the myoglobin. So looking at these graphs again, at any given partial pressure of oxygen, the oxygen is picked up by the adult hemoglobin at a certain pressure. It then gives up its oxygen to the fetal hemoglobin, which is better at picking up oxygen at that same partial pressure. And then the oxygen is given up to the myoglobin, which is even more efficient at picking up oxygen from either the adult or the fetal hemoglobin. The evolution of the globin gene family is fairly complex. It's been studied in great detail. Starting with the placental mammals, there are quite a few different versions of the alphas and the beta genes because of some of the reasons I just stated earlier. Going back to marsupials, they have fewer copies of the alpha and beta globin genes, primarily because they're different developmental pathway. Sometime around 300 million years ago or so, there was a separation on different chromosomes, and then about 450 to 500 million years ago was the initial gene duplication that formed the alpha family and the beta family invertebrates, and those were primarily in the jawless fish and eventually in the jawed fish. And then going back a little further, you have the progenote gene, which was essentially myoglobin. So looking at it forward, first there was the progenote gene in invertebrates. As these were inherited by the vertebrates, there was a duplication, and actually several duplications, followed by separation onto separate chromosomes in the jawless and jawed fish. Then there were further duplications and diversifications, first in the marsupials, which the embryo would then come out of the mother, so there would be different oxygen requirements. But then in the placentals, where the embryo was held for, and the fetus were held for extended periods of time inside the mother, inside the womb, then different methods of oxygen transfer were required and further diversification of the globin genes. When the globin genes were first discovered, then they discovered that there were pseudogenes involved. So there are different types of pseudogenes, both expressed pseudogenes and non-expressed pseudogenes. So the first type, if you look at the functional gene, the promoter region has changed. So the gene essentially is intact, although the promoter is faulty. The second type is forms a stop codon or a nonsense codon, so that leads to a lethal change for that protein. Another type is just a truncated gene. It only has a portion of the gene. And then there's a processed gene that has no introns, but it does have a poly A region. So that's another type of pseudogene. Now, as I stated earlier, some pseudogenes are never expressed as RNA or a protein. There are other pseudogenes that are actually expressed as RNA and proteins, although the proteins are faulty, and those are also pseudogenes. Histone genes in eukaryotes also make up a multi-gene family, and they're shown above as H1, H2A, H2B, H3, and H4. And they evolved out of a group from the archaea down below, the archaeal types. The first separation was between an H3, H4 group and an H1, H2A, H2B group. And then there was further diversification which split the H3 and H4 group, as well as the H2A, H2B, and H1 group. And finally, a final diversification that led to a separation of the H2B histones and the H1 histone genes. In the majority of cases, most of the gene duplications and therefore the expansion of these multi-gene families originated from genome duplications, that is polyploidization. So you have a duplication of the genome, it duplicates all the genes. Now you have at least 2 copies of each one of the genes. You can get another duplication and have four sets of those genes, and then they can start diversifying. And this has actually led to a lot of diversification of not only the genes, but also the organisms. In the case of this phylogram that I'm showing up here, each one of the little circles represents a genome duplication event. So If you go from in the animals up at the top, you have a gene duplication before the diversification of vertebrates, and another duplication after that to split the mammals, etc., off from the fish. And the fish, actually the teleosts, the ray fin fish, had an additional duplication. So you go from a set of diploid organisms into tetraploid into octoploid, and then they have 8 copies of each one of the original genes, which can then diversify, and they have. So in the case of plants, which are shown on the right-hand side, there have been multiple genome duplication events starting in gymnosperms, in ferns, in magnoliids, in monocots, and in eudicots. And each one of those is an underestimate, actually, of what's actually happened. Since I produced this picture, many more gene duplication events have been noted in different plant species and different plant groups. As an example, Hox genes, which are homeotic genes that control developmental processes, if you look at an ancestral chordate at the top, they had a certain set of Hox genes, 14 different ones. And then if you go into jawless fish, what you find is they have 2 copies. And if you look at other genes, it's clear that there's been a genome duplication event. And in this case, then you get diversification so that some of those gene copies mutate to the point where they're no longer active anymore. In cartilaginous fish, that is sharks and rays, you have a further duplication event to form 4 copies of those genes, and again you get diversification of the gene copies from that. And then in ray fin or bony fish, there have been even further duplications, such that you have 8 copies, and in some cases a whole locus goes blank. But in general, each one of the genes is free to mutate, and that's how you get this diversification in body type, body style, where the arms are formed, where the fins are formed, etc. So in this figure, a phylogenetic tree at the bottom is relating the number of copies of those Hox genes, same colors used here as in the previous slide. Starting with an ancestral chordate with all 14 of those Hox genes in order, and then a genome duplication to get the jawless fish, and already some evolution happening as some of the genes are blinking out or going extinct, essentially. And then a further duplication to form the jawed fish pattern, including sharks, which are the cartilaginous fish. And then up to the ray-finned fish up there, where you get a further duplication. And zebrafish and pufferfish actually have different versions of those. In one of the C loci is turned off completely, and in the other one, the D locus, one of the D loci is turned off completely. As you work up further, the lobe-finned fish have only undergone 2 genome duplication events, as have mammals and birds, and you can see the different patterns in each one of those to form each of the different body types. On the top, you can see the changes in some of those Hox genes. The Hox D13 is that one that's in the D line, number 13. And as it increases and increases over the hundreds of millions of years, the number of digits on those appendages becomes less and less, and the digits become more and more pronounced. So you go from having a fin to a lobed fin to eventually having toes and fingers. And those are all caused by changes in those Hox genes. I will say a bit more about these in the next presentation as well, and in a later presentation I'll add a few more details. Okay, next I want to talk about multicellularity and cell communication, both of which are somewhat related to the topic I've just been talking about. So much of this next section comes from chapter 16 in the Integrated Molecular Evolution book. This is a phylogram showing all known cellular groups, including bacteria, archaea, excavata, microbians, rhizaria, alveolata, stromatophiles, archaeplastida or plants, amoebozoa, the opisthokonta, which has the fungi and the animals within it. All of the lighter areas, the white areas, indicate the amount or proportion of unicellular representatives within that group, and all the dark areas indicate all of the multicellular organisms within each of the groups. So you can see, even with bacteria, certainly most of them are unicellular, but there are a few that are multicellular. The same with archaea, excavata, and some of the protists, although stromatophiles have a lot of multicellular representatives. By the time you get up to plants, amoebozoas, and opisthokonts, most of the members are multicellular. If you just focus in on the Animalia clade, you can see most organisms are multicellular, although at the base, down here, are single-celled organisms called choanoflagellates. Although a lot of choanoflagellates tend to aggregate, and so they're sort of on their way to multicellularity. As you work your way up here, there are cnidaria, which are the anemones, like sea anemones, and then a split off here to the echinoderms and chordates, including tunicates and, of course, the vertebrates up here. Over here are things like annelids, mollusks, brachiopods, and nematodes, and finally up to arachnids, insects, and crustaceans. In multicellular organisms, the cells have to be able to communicate. Otherwise, you have complete disorder. They won't be able to communicate, and you'll have things like tumors and cancers created. So there are a couple of different types of signaling in cell. One set are called paracrine signaling mechanisms, where adjacent cells or cells in the same vicinity are communicating with one another. They connect up, they secrete different chemicals in order to communicate with one another, and this happens a lot in the immune system. There's also signaling with neurons, that is, one neuron sends an action potential into another neuron to send a certain message to either stimulate that neuron or to calm it down, essentially. There's also long-range signaling, such as endocrine signaling, where you have chemicals released by one cell then go through the bloodstream and affect a whole set of cells in a different part of the body, such as insulin responses, adrenaline responses, etc. Although these signaling systems are associated with multicellular organisms, they had their beginnings in unicellular bacteria. In bacteria, one of the ways that they sense each other is something called quorum sensing. So they will create certain molecules, inducer molecules, that at low concentrations won't stimulate themselves or any other bacterium in the same area. But at higher concentrations of bacteria, when more of those molecules are around, they'll enter the cell, interact with a receptor, which will then cause the increased transcription of target genes and gene products. And sometimes these gene products emit light, so you can see these things fluorescing in the ocean and other places. Another type on the bottom is when the inducer is a different type of molecule, more of a hormone type. And at low concentrations, again, nothing happens, but at high concentrations, it will interact with a receptor on the membrane, a transmembrane protein, which will then send a signal inside the cell to activate a transcription factor and promoter, which will then activate those gene products. So here are a few examples of that. In one case on the upper left, there are bacteria that form a type of lactone, which is then released from those cells. It can enter that same cell, but at low concentrations it normally doesn't. At high concentrations, it will interact with other cells, interact with a regulator protein, which will then act as a transcription factor to turn on genes or express some genes. On the upper right, signal peptides are produced that are then exported using ABC transporters, and then those interact with usually another cell, a receptor on another cell, that changes a protein within the cell, phosphorylates it usually, activating it, and then it'll interact with a response element that will act as a transcription factor to increase the transcription of genes and expression of those genes. Then on the lower part, there's one that's a little more complex in that several different molecules are produced that interact with different receptors, some of which can stimulate expression, while others will actually inhibit expression of the target genes. And in this case, the target genes produce luciferase, which produces a flash of light. And so you can see the ocean waves and out in the ocean fluorescing blues and greens based on the amount of bacteria and the amount of those chemicals and hormones that they are releasing from those cells. So bacteria are capable of signaling one another. That means that bacteria can exhibit a number of different expression profiles. And in particular, some bacteria that produce more than one cell type can have different expression levels. And one of those is Caulobacter crescentus. And in this case, there are two different types of cells. There are swarmer cells and there are stalked cells. The swarmer cells swim around until they find a spot where they deem it's a good spot for reproducing. And when they do that, they will then send out a stalk, and the stalk will stick to the substrate below, whatever that happens to be, and it'll be there forever. So it'll never come loose from that spot again, at least in its lifetime. Once it does that, it'll start to divide, and once the cells start to divide, they start to differentiate. So the two different cells would be a stalk cell and a swarmer cell. The swarmer cell would start developing a flagellum, and ultimately, when the two cells separate, the swarmer cell can then swim away and find another spot to settle down in. Once it settles down, then it gets rid of its flagellum and starts growing a stalk and repeats the process over again. Plants and animals have primarily multicellular representatives, although there are a few unicellular representatives, including some green algae, red algae, and in animals, choanoflagellates. But about 1 billion years ago, they started aggregating and then there was cell differentiation. So they were communicating with one another. So the first group then started just aggregating, then others aggregated and had specialized cells. Eventually they had specialized cells and new colonies which originated from inner cells as well, so they were reproducing in a different way. So as far as Chlamydomonas, it's a unicellular organism that's is motile, and then you have gonium, where the cells aggregate and communicate with one another to form a little jelly-like structure around them. And then Pandorina, which forms sort of a tiny little ball that is also motile with all the flagella pointed outwards. Next is Euderina, which also forms a slightly larger ball and is reactive to, again, sunlight, so they have eye spots that can cause the entire organism to move towards the sunlight. Then there's Pleoderina, which has some large cells, some small cells. The small cells are light reactive so that they can move around, and the larger cells do most of the photosynthesis. And then finally things like Volvox, where you have small cells around the perimeter forming a ball, which is then motile. They have eye spots so they can see where the light is coming from. And then internal larger structures, which are actually also multicellular and eventually break out to form new colonies. Animals also have similar evolutionary patterns as far as going from single cell to multicellular organisms. And as I've already mentioned, the Hox genes and pair rule genes and other types of genes have a big part in forming body plans in different organisms. By the time you get to things like sharks, which are, fish essentially, lizards, chickens, pigs, and humans, even there, on such divergent vertebrate species, you have some very common mechanisms and developmental mechanisms to produce the final organism. In fact, there's a vertebrate set of genes, essentially, that sets down the basic body plan, and then there are variable genes after that that lead to the more of a differentiation between, say, a lizard and a human, or a shark and a human. Interestingly, some of the genes that are involved in development of animal cells also are involved in cancer development, and so I'll be talking about that in a couple of lectures from now. But just to introduce it at this point, there are growth factors, external membrane receptors, and then internal intracellular messengers and transcription factors, as well as things like cell proliferation controllers, cell damage regulators, and apoptosis, or programmed cell death regulators, that are involved in multicellularity and development of these cells in reaction from one cell to the other. But they're also involved in the problems that go on when some of these genes go wrong and eventually cause cancer to develop. This slide presents it in more of a succinct way. So at the top you have hormones and different molecules that affect cancer and cell-to-cell recognition. And then there are receptors on those that recognize those particular compounds and cell-to-cell contacts. Inside of that, within the cell, are cell transduction regulators. And so if you have the signal coming into the cell but not being recognized properly, that can cause problems in the cell. And then you have gene expression control, that is mainly transcription factors that cause problems in cancer. Cell cycle control, that is controlling when or when not to have the cell undergo replication and division. Cell damage regulators, including BRCA1 and BRCA2, which are breast cancer genes that are primarily there to detect DNA damage. And then apoptosis, or programmed cell death regulators that determine when there's too much damage to correct, then it's better to kill the cell. So there's a great deal of coordination and communication among the cells of a multicellular organism in order to assure that the entire organism can survive to reproduction. Even prior to fertilization, the egg cells of animals and plants, for that matter, have differentiation as far as one end of the cell versus the other end of the cell. So in the case of animals, there's an animal pole and vegetal pole. It just means that there's a difference in the gene expression and the protein content of each end of the cell. So once fertilization happens, there's rapid cell division, and then eventually those cells form what's called a blastocele, which is a hollow area within that developing embryo, followed by the invagination of a different area that will form another hollow area, and the blastocele will actually shrink, eventually forming several types of tissue due to interaction of the different cell types. So there's an ectoderm, which will essentially form the outer portions of the organism, as well as the interior of the gut, as well as some neuronal tissue. The endoderm, which will form much of the internal organs, and then the mesoderm, which is an interactive group of cells that reacts to signals sent by the ectoderm and the endoderm. In some cases, this interaction between the endoderm and ectoderm causes some of the mesoderm to become secondary mesenchyme. These are cells that tend to move around. They also tend to interact with other cells. One of the cell types are melanocytes. These are cells that form pigment that winds up in skin, but it doesn't originate in the skin cells. Because they move around, that if these cells eventually become cancerous, they tend to again move around just like they did in the developmental stage, and so they are ones that can metastasize readily and early in the course of this cancer. I will talk about this a little later in a cancer lecture as well. As far as where these different cell types come from, skin cells, as well as neuronal cells and some pigmented cells, arise from the ectoderm, whereas for the endoderm, these are primarily pancreatic cells, thyroid cells, lung cell progenitors. And then the mesoderm, which is interaction between those two, those become cardiac muscle, tubule cells, red blood cells, and other types of cells. And then there are sperm and egg cells. These are actually set aside very early during development so that they don't undergo too many cell division. Here are some of the developmental pathways for blood cell development. Starting with multipotential hemopoietic stem cells. Those will divide to become either myeloid progenitors or lymphoid progenitors. The myeloid progenitors can become megakaryocytes, erythrocytes, that is red blood cells, mast cells, or myeloblasts. And the myeloblasts can further differentiate into basophils, neutrophils, eosinophils, and monocytes, and the monocytes can progress into macrophages. These can be involved in oxygen transport, such as erythrocytes, clotting for the megakaryocytes, as well as the innate response, immune response. Those are the basophils, neutrophils, eosinophils, and macrophages. As far as the lymphoid progenitors, those can then progress, differentiate into natural killer cells or lymphocytes, and those lymphocytes can become either B cells or T cells, and there are a number of different types of T cells. And B cells can further differentiate into plasma cells. A lot of different differentiation starting from just one cell type, and that's just by changing and reacting to different conditions and changing the expression of certain genes in each one of those cell types. Now, to move up to a complete organism, one of the ones that's been studied the most is Caenorhabditis elegans, which is a nematode. It contains about 1,000 cells, and so they're easy to follow through. Caenorhabditis elegans has males and hermaphrodites. There are no true females, so the hermaphrodites can fertilize their own eggs and give rise to new individuals. Here on the bottom you can see the developmental pathway for part of the development of Caenorhabditis elegans. If you were to look at the human pathway during development, it would look very similar. Here are the two organisms. In essence, it's primarily a mouth, a gut, and sexual organs. The two sexes in Caenorhabditis elegans, the hermaphrodite that has exactly 959 somatic cells, a third of which are neurons, and sperm and egg, and the male has 1,031 somatic cells, again about a third being neurons and sperm. They have mouth, pharynx, intestines, rectum, and anus, so they can eat and digest food, and then they have sexual organs. The female has both a gonad for producing oocytes and also spermacia for producing sperm, while the male has only a gonad that produces sperm in a seminal vesicle. Approximately 1 out of 1,000 individuals is a male, so there aren't many males, and so most of the reproduction is done by the hermaphrodite. In some species of Caenorhabditis, there are no males, and so all of the reproduction is done by the hermaphrodite. So researchers for many years studied Caenorhabditis elegans, especially following the cell divisions in each case during development. And what they found was at certain points in development, during some cell divisions, one cell always died and one cell always lived. And so there were cell fates, that is, during some cell divisions, one cell was always destined to live and one cell was always destined to die. And this was then termed programmed cell death, or apoptosis, for the cells that would die normally during a developmental process. You can see this in the diagram, in that in some of the cell divisions you have red, blue, and green arrows indicating cells that are destined to die during that developmental process. Here is one example of that, which makes perfect sense. So the male chemotaxis pathway. Males, of course, have to find a female, and so they need to be able to not only sense where a female is, but then move towards that. So they need neurons that will actually allow them to sniff out essentially a female and then move towards her. So in a male, during a certain cell division, the CEM cell develops normally, whereas in a hermaphrodite, which does not need to find a female, that cell dies. There's programmed cell death. Conversely, in a hermaphrodite, the hermaphrodites lay eggs, so they need to have a specific neuron set that allows them to lay eggs, that is, sense when the egg is ready and then use the muscles to extrude the egg to lay it. The males don't need that. They don't have eggs, they don't need those muscles, they don't need those neurons. So in a certain cell division, that particular cell dies, and those pathways and those cells are not produced in the male. So again, this is an example of programmed cell death or apoptosis. So that is the end of this section. I will continue in the next lecture on multicellularity, primarily focusing on animals, but also having a fairly large section on plants. And that will come from chapters 16 and 17 from the Integrated Molecular Evolution 2nd edition. Okay, thank you.